Alright, so let's look at rhodopsin-retinal visual cycle. We already know that from the internal and external structure of the eye, that as light passes through the outermost is what? The outermost covering transparent is what? Cornea, right? And the light goes through the way. Movie, right? And it's on the what? Lens, right? The lens is now the back light, right? Because it's a what? So an inverted ring image will form on the what? Retina. Now we say information from the retina will be sent to the brain, right? But how is that done? That is what we want to do. We will send through the optic nerve to the brain. How is that done? Now this is how it is done. We said in the retina we have two main types of cells, right? Receptors, which are what? Rod cells and what? Cones. Now we want to see how cone is involved, the rod is involved in vision. Now, the rod contains a light-sensitive pigment called what? Rhodopsin. And we say that rhodopsin is a combination of two things, what and what? Rhodopsin and what? And 11-6 what? 11-6 retinal. Now, this is our rhodopsin. So you already know that this rhodopsin here, in the rod cell, in that retina, is rhodopsin plus 11-6 retinal, right? Now, as light, that light from the lens hits the rod cell in the retina, hits this rhodopsin. The rhodopsin will begin to dissociate. To break down. Do you get? And the particular part of the rhodopsin breaking down is the retinal part. So the retina will be converted to, the 11-6 retina will be converted to what? All trans retina. I take that again. As light strikes on the rhodopsin, it will begin to what? Decompose. And the part of it decomposing is the retinal part of it. Remember, it contains rhodopsin and retina. It is the retinal part of it that is decomposing. That 11-6 retina that was binding to rhodopsin will be converted to what? All trans retina. But remember that all trans retina cannot bind to rhodopsin, right? So because the 11-6 retina have not been converted to all trans retina, they can no longer bind together. And so the all trans retina will begin to dissociate from the rhodopsin. Are we getting it? We begin to dissociate, fall out from the rhodopsin, and we form the first product, rhodopsin. In rhodopsin, the all trans retina are not completely falling out, partially dissociated. Do you get that? Rhodopsin. And we said in nanoseconds, this is converted to what? Looking rhodopsin, right? As the dissociation goes on, like as they are separating from each other, it's a gradual process. The all trans is gradually separating from its rhodopsin, gradually. In microseconds, rhodopsin is converted to what? Metal rhodopsin 1. Now, slowly now, metal rhodopsin 1, in microseconds will be converted to what? Metal rhodopsin 2 is a very small process, 10 seconds. Metal rhodopsin 2. Now, this metal rhodopsin 2 now can finally dissociate into, it's a metal rhodopsin 2 is also called what? Activated what? Rhodopsin, right? Activated rhodopsin. It is from metal rhodopsin 2 that the all trans retinal will completely dissociate from the rhodopsin. So at this point, they are finally dissociated. Our all trans retinal is now here. Our rhodopsin is now here. All trans retinal, rhodopsin, dissociated from each other. Now, it is this metal rhodopsin 2 that sends signal into the rod cells that when we are saying that the retina sends signal to the optic nerves. Read the last part I gave there. From metal rhodopsin 2. What did I say?
Also called
excites electrical charges in the rods, right?
Yes.
That does what?
And the rod then transmit
the information to what? The information to the central nervous system. So it is this metal rhodopsin 2 that excites charges, electrical charges in the rod cells. As electrical charges are excited in the rod cell, impulses now be sent where? The what? The central nervous system. Already you know that the central nervous system consists of what? The brain and the what? So we are talking about the brain here. Now, so aside that, this rhodopsin here can be used to reform rhodopsin. Remember, rhodopsin is just rhodopsin plus what? 11 cis-retinal, right? So if we can get 11 cis-retinal from somewhere, which we will see, then the 11 cis-retinal can combine with rhodopsin to reform what? Rhodopsin. Is it not? Now, this all trans-retinal can be converted to all trans-retinol. This retinal, RDI functional group. This retinol, alcohol. So it can be converted to all trans-retinol. And all trans-retinol, you can tell me what is what? A form of vitamin A, right? Yes. Vitamin A. We are coming to when they say vitamin A is good for your eye. How is it good for your eye? We are gradually getting there. Now, the all trans-retinol now can be isomerized to what? 11-cis. So on position 11, that trans bond there can be converted to a cis bond. Good. So it can be isomerized to 11-cis retinol. And 11-cis retinol can be converted back to what? 11-cis retinol. 11-cis retinol. So the 11-cis retinol can combine with rhodopsin to form rhodopsin. And the rhodopsin becomes stable, waiting for another light to excite it. Do you get? Let me summarize that again. We are looking at the rhodopsin retinal visual cycle. How do we see? As light passes through the cornea, the what? The pupil hits the what? Lens, gets onto the retina. Now, what part of the retina are we talking about? Now, we said the light sensitive pigments in the retina are the rod cells and the bone cells. But now we are taking the rod cells first. The rod cells, we say the light sensitive pigment in the rod cell is what? Is what? Rhodopsin, right? Rhodopsin, good. Now, that rhodopsin is made up of two things. We said it's made up of what? Rhodopsin and what? 11-cis-retinal. This is what makes up this rhodopsin. But as that light strikes the rhodopsin, it will begin to dissociate. It will begin to this 11-cis-retinal, as the light strikes this, will be converted to be isomerized to all trans-retinal. That is, cis bond will be converted to the trans one. So we now have all trans-retinal. Do you get it? And because all trans-retinal cannot be fused to the rhodopsin, they will begin to dissociate because the three-dimensional structure of this is different from the active side of this. So they will begin to dissociate from each other. Now, that dissociation will cause the first intermediate here, photoreceptor. That is, photoreceptor is from when the all trans-retinal now is partially dissociated from the rhodopsin. This is very unstable. And it will be converted to rhodopsin, to metal rhodopsin 1 and metal rhodopsin 2. Now it is this metal rhodopsin 2 here that will cause the what? That will cause what? Excitement of what? Electrical charge, electric charge. And this charge now will not send what? Will not stimulate what? The rod cells, right? Send impulse. Impulse now will not be sent to the central nervous system. But this metal rhodopsin 2, which is also called activated rhodopsin, can dissociate finally now to our all trans-retinal and our rhodopsin. This rhodopsin can be used to reform rhodopsin, or and the all trans-retinal can be converted to all trans-retinal. They can be converted actually. This can be isomerized to 11-cis-retinal. 11-cis-retinal can be converted to 11-cis-retinal, which can then combine with rhodopsin again to reform this rhodopsin. The rhodopsin is stable and waits for light into it again and before it dissociates. Do we get that? So if you are asked to explain the rhodopsin-retinal visual cycle, this is what you have to explain. Yes.
From that all translate, and it goes straight since we have an arrow.
Can't it go straight to where?
To the 11 to the retina, since it's to the right.
Like this.
So all translate yes.
To what?
11 to the retina. Since the arrow is
11 is what?
retina.
It's going straight now.
And based on what you expect, this one was converted to all translate then to 11 to the retina. I'm asking, can't you just go straight?
No, it's going straight. But what if we get all transretinal? If we have all transretinal, like in the case of the vitamin A, how does it aid eyesight? We come there, that's I'm not going to explain because we get there. Any other question? Any other question? Yes. Yes, you draw it in exam. This is pathway. Sorry.